from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications but also contribute to data that is used for translation for better public health.
Visiting lecturers or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork and very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Happy 10 years, WACCBIP.
Happy 10th anniversary, WACCBIP.
Happy 10 years to WACCBIP.
Happy 10 years, WACCBIP.
happy, glorious, and illustrious 10th anniversary, WACCBIP.
Happy 10th anniversary, WACCBIP.
Many congratulations to everybody involved in the WACCBIP story.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had, and their desire to learn, and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these.
We're getting ready for the next half of the day, the business end. And if you have friends outside, send them a message and just let them know that we are getting ready to set off. We have a long day ahead of us, so the sooner we start.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of the department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up. So I called Gordon and said, Gordon, I think you should host this.
So basically I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Teresa, Osborne, Abidad. There was another lady called Marian Yaku, the vice chancellor at the time, Professor Ernest Aite. I was determined to turn the university.
I hope we're all refreshed and ready for the next session. Before we start with our keynote lecture, it was mentioned at the beginning in the opening session, but one of the key partners that we have within the government infrastructure is the National Vaccine Institute. And we are honored to have the Deputy CEO of the National Vaccine Institute here. They were at a function in Ada and only just got here. So he'll say a few words before we introduce our keynote and get along with the scientific program. So Dr. Nuhu.
Thank you very much. Good morning, everyone. I tried to get here early. I didn't know we could have traffic that heavy. But however, I'm here. My boss, Dr. Sozi, he wanted to be here, but he has engagement somewhere. So I'm here today. Tomorrow he will surely join us. WACCBIP, we all know that it stands at the forefront of biomedical research in Africa. And we also know that WACCBIP is helping to build capacity, produce cutting-edge research, foster collaborations, not just in Ghana, not just in Africa, but collaborations that spans the globe. Today I can tell you, because of WACCBIP, that we are seeing vibrant, growing community of African scientists tackling infectious diseases through rigorous inquiry and advanced technology. In short, we are seeing African scientists working to solve African problems. This year's theme, Evolving African Science from Capacity Building to Discovery and Impact, at least to MVI, could not have been coined any better, because it's as if it's speaking to the core mandate of MVI: capacity building, discovery, and impact. So over the coming days, we will hear from leading scientists like yourself, scientists that are working in areas like cell biology, immunology, and bioinformatics. We will witness brilliance of early career scientists engage in critical conversations and exploring new frontiers never explored before. So I would like to take this opportunity, because I understand research don't just happen. Research is backed by funding and finance. So thank you very much, our development partners and collaborators. And to our students and your research scientists, you are the future. And I'm also conscious you are part of the present. So let this conference inspire you to keep asking hard questions and to keep pushing boundaries, to remain anchored in excellence and purpose, for we know that you are resolute. Thank you very much.
Thank you very much, Dr. Nuhu. That's Deputy CEO of the National Vaccine Institute. And it's time for our first keynote lecture of the conference. It's my distinct pleasure to welcome one of our collaborators, a very powerful scientist, a senior clinical research fellow at the Francis Crick Institute and the University College London, Hospitals Biomedical Research Center. And she's about to start her own research program at Queen Mary. I'm not sure if I'm supposed to share that, but I thought I would. And she's a consultant in infectious disease and acute medicine at URCH. She's an academic consultant in infectious diseases at University College London and a clinical research group leader at the Francis Crick Institute. She also leads what has become one of the more powerful COVID-19 prospective cohorts, the Legacy study. And she's a collaborator on our West Africa, West Indies, West London Consortium, WWW Consortium. And I believe some of what she's going to talk about will touch along that. Dr. Wall graduated from University of Bristol in 2000 and then did postgraduate training and actually got her PhD at the Liverpool School of Tropical Medicine and the Malawi-Liverpool Wellcome Trust Clinical Research Program in Malawi. So she's a bona fide African and very well placed, considering these times and the capacity that she's built and the research outputs that she's built in this during COVID and post-COVID world, to lead off as our first keynote lecture of WRC 2025. So Dr. Emma Wall, please come and give us your address. Thank you.
Hi everyone, it's an enormous pleasure and honor to be here. Thank you, Gordon, for the invitation. Hopefully my slides will appear in a second, but I'm a very unusual beast. I am a clinician with a foot in the hospital, the patients, the clinic, and another foot in the research centre, which is the Francis Crick Institute, which is essentially founded around the principles of understanding cell biology. So it's like a giant WACCBIP on steroids, with a few Nobel laureates in the building. But essentially, the Crick was designed to understand cell biology and is full of really basic scientists, and they have no idea how to, in many of them, translate those results into patient care, into better medicines, into better treatment. And most of my colleagues in the hospital have no idea what happens in the Crick and what people do. And so part of my job is linking up the two, getting people to talk to each other, making sure that we do the best possible science. And I've lost my slide advancer. Is it that one? There was another one. Thanks. So, as Peter mentioned, I've done a year, working in Mbarara in Uganda, which gave me a passion for research and medicine in Africa. And then I went and did my PhD in Malawi for 4 years at MLW. Many of you will be familiar, and shout out to Malawians who've made it here. And then I've been involved in... You want to check this working? Oh yeah, it's going out. Which gave me a passion for Thailand, but then Wuhan placed under quarantine, 10 days later. The virus had already gone into Thailand too. As COVID emerged in London, it was very clear that there was no testing available anywhere. And so the Crick did an extraordinary thing. Instead of being able to just shut down like lots of other research institutes and do not very much, it turned its entire scientific staff and facilities into a giant PCR testing pipeline, and provided occupational health testing for healthcare workers, care home workers, and research institutes for 2 years across northwest London. And it was a partnership between these two organisations, and this is how I got involved, because I come from UCLH, and I happened to be at the Crick at the time. And it was linked by a company that provides testing services. And essentially, over 2 years, hopefully this is going to work, the pipeline conducted 680,000 PCR tests with a median turnaround time of 8 hours across 12 NHS hospitals, 98 local care homes. I have to point it over there, do I? Okay, 1 mental health trust and 4 research institutes. And what this meant was that to go into the Crick, you had to have a negative PCR test to get entry, which meant we could continue safe research. And people who tested positive had to stay home and isolate. And so this is a map of London, and essentially we were able to cover a huge swathe of London, healthcare workers in London, who were being highly exposed to SARS-CoV-2. And we conducted an extraordinary number of tests. You can see a wave for alpha there and a wave for the original positive D614G strain there. And we captured all of the positive tests and all of those healthcare workers sent home. And what we were able to do, therefore, is go back and look at the data and show and model how many people or how many transmission events in the hospital we've been able to avert. When we looked at the actual variants that we had found, unsurprisingly, all of the variants here in proportion and time on the x-axis here were exactly the same as the rest of the country experienced, unsurprisingly. But when we went back into the data in a bit more detail, we found that alpha, we first detected alpha in healthcare workers a month before it was declared a variant of concern by the WHO, and delta 3 weeks before, Omicron, people were on the case by then. But the healthcare workers were a canary in the coal mine. They were a population where surveillance was incredibly important, not only to keep them infected people out of the workforce and stop transmission events to patients who needed ongoing, particularly surgical and cancer care, but to make sure that you can detect early variants as they're circulating and as everything's changing. So I'm going to switch tack a tiny bit now and start talking about vaccines. Here's the famous New England Journal paper. I still think this is one of the most extraordinary Kaplan-Meier curves I've ever seen of vaccine efficacy. It is one of the most extraordinary Kaplan-Meier curves I've ever seen of vaccine efficacy in the trial conducted of the Pfizer-BioNTech vaccine in 2020. Here's the cases in and here's the people who received placebo. Extraordinarily effective vaccine. And what became clear very quickly was that the vaccine efficacy from these vaccines was directly correlated with the neutralization level in serum, neutralizing antibodies in serum. And this was further confirmed by the recovery trial, which was a large platform trial in the UK. You may have heard about it, conducted across virtually every hospital in the country, testing interventions to reduce mortality. This is part of the trial that was looking at a neutralizing antibody cocktail called Regeneron. And essentially for people who are antibody naive, if they received Regeneron, their mortality was lower. If they were antibody positive already, they didn't have any difference in their mortality. So showing again another piece of evidence that neutralizing antibodies are crucial in mediating mortality from COVID-19. So where do I come into all of this? Well, that giant testing pipeline I talked about, as it was ramping up and functioning, the vaccines were coming on board, and the leadership of the CRIC said, we're a research institute, how do we operationalize this for research? Testing pipeline I talked about, as it was ramping up and functioning, the vaccines were coming on board, and the leadership of the CRIC said, we're a research institute, how do we operationalize this for research? And so they created the colleague of mine, so I can WhatsApp him and tell him that some data's on the way. That's always been useful, but overall, we, in the infectious diseases community in the UK, we're well networked. So it's fairly straightforward for all of my colleagues to feed data into policymakers. We've also been feeding data into the WHO for vaccine strain selection. And out of this, we've had a lot of papers and a lot of outputs which have been cited a lot in policy because we're able to answer questions quickly. So here's what the cohort looks like. This is up to the end of 23, and essentially we've got every vaccine and every bleed time point. And overall, we can see here that this goes up to XBB 1.5. I'm sorry, my colleagues at the back are doing a great job keeping up with my pointer. That the breadth of neutralizing antibodies across the cohort has increased significantly from this very early time point when we get to 2023, when actually nearly everyone neutralizes everything. But it's taken time to develop. And what we're able to map this to is seroconversion rates to N and the exact variant that people were infected for, because our cohort remains under active PCR surveillance. So we've got a number of research platforms. I don't have time to talk about them all today, but essentially I've talked already about our rapid humoral immunity platform, which involves both healthy and immunocompromised adults from the UCLH BRC. We're building up a piece of work around mucosal immunity that's developing very nicely. We look specifically at infections and looking at how different variants cause different infection severity with human samples and animal models. We've been running a program testing therapeutic antibodies for COVID-19, whether they still neutralize the latest variant, and feeding that data into policy, and particularly the National Institute for Care Excellence, and this modeling program, and I'll talk a bit more about that. So the first question we were asked in 2021 was, Delta's on the way, is our vaccine going to protect us? And this is really what is extraordinary, what we found. There are two vaccines being used in the UK, the Oxford AstraZeneca vaccine, an adenovirus vector vaccine, and the Pfizer-BioNTech mRNA vaccine. And after 2 doses, across all the variants we tested, that there were neutralizing antibodies detected. But you can see, particularly here against Delta, there's a real split in the AstraZeneca recipients between these people who have detectable antibody, the log2 scale, and people who don't. So what drove that? I think you can all guess. Oh, it was infection. So people, at that time we didn't have an N assay in the building, so we were asking people, did they have prior symptoms? No, no antibodies against Delta. Prior symptoms, yes, lots of antibodies, well not lots, but detectable antibodies, showing that infection was priming that vaccine response and that breadth of vaccine response. As we move forward, this became really apparent, and to the point where leadership of the CRIC were being approached by senior scientists in the building saying, I don't think Emma and her team know what they're doing because they keep finding all these funny antibodies that people shouldn't have. So this was before, these people were bled before Omicron turned up. There was no Omicron at the time in the UK, but everyone was being offered a booster vaccine or third dose of vaccine and were being bled. And actually half of them had pre-existing anti-Omicron neutralizing antibodies. They got a third dose of ancestral monovalent vaccine, and they got a massive boost. And this idea that actually there were antibodies being produced against variants that didn't exist yet. It challenged a lot of the notions in immunology that you develop a response to the antigen you see rather than an antigenically distinct antigen. And over time, we've seen the same thing over and over and over again. So here now people are getting their fourth dose. This is a bivalent vaccine containing Omicron BA1, but lo and behold, oh, sorry, I'm not doing very well with this. People were developing and getting already, lo and behold, oh, sorry, I'm not doing very well with this. People were developing and getting already had and were getting boosted against XBB 1.5, a variant that didn't emerge for another 12 months. But what was surprising is this variant hadn't yet emerged, and yet both vaccines boosted it really nicely. And this was the mama of JN1. Vaccines boosted it really nicely. Vaccines boosted it really nicely. And this was the mama of JN1, which is then the mama of all boost. We know that based on last year's data. So you're using trying to interpret last year's based on last year's data. So you're using trying to interpret last year's bleeds against current variants to try and predict whether that vaccine will generate bleeds against current variants to try and predict whether that vaccine will generate protection in the future. That's a difficult thing to do. We'll predict whether that vaccine will generate protection in the future. That's a difficult thing to do. Wouldn't it be nice if we could model where the virus is going, where the virus is going, where the immune system is going, and design a vaccine ahead of evolution? Well, I'm part of a UKRI-funded £8 million consortium to do exactly that. And we're trying to figure out how does the infecting variant influence the antibody boost we get from a vaccine? There we go. And so this is some of our legacy data drawn out, and we chose 3 peaks. We chose a Delta wave, a BA2 wave, and an XBB 1.5 wave. And that's because these were well-defined waves in the UK where we knew exactly when the virus came and when it finished, when it went away. And we also knew when our participants had been exposed through our PCR surveillance, and we also knew when they'd been vaccinated. So we could use this as a really robust data set on which to build the model. And what we found, essentially, we looked at the ratio between the population level titer at the set point and the peak across our 3 waves, Delta, BA2, and XBB. And what we're looking at here is people with a round circle who were uninfected and people with triangle who had an infection. And you can see already, even in the Delta wave, if you'd had an infection, you were boosted against Delta, and if you had an infection, you were more boosted against Omicron. And then when we move into the BA2 wave, we're able to add in a pre-Omicron or even in the Delta wave, if you'd had an infection, you were boosted against Delta, and if you had an infection, you were more boosted against Omicron. And then we look at the virus and put into antigenic space the different variants and see if they cluster by predictive mutations in spike that we think are driving antigenic diversity. And this is very nice modelling to do in a hamster. You can give a hamster a very unique single exposure and you can see where that variant you've infected your hamster with lies in antigenic space. This, however, gets much messier when we start to put it onto the human data. And this is our 5th dose cohort, pre-5th dose and post. And I've labelled the variants here, and you can see that some of the variants that emerged, JN1, BQ1.1, they're starting to look antigenically distant from the cluster in the centre, but the moment everyone gets a boost, another exposure, they all start clustering together again. So how useful antigenic cartography is for mapping out the variants and how they're changing in a very heterogeneous exposed population compared to a hamster is difficult. And one of the things we're trying to resolve is how do we look at the virus and look at the immune response, because actually the virus is evolving, the immune response is evolving, it's like a giant hamster wheel that keeps. Yes, that while we continue to see antigenic reassortment, actually our current vaccine strategy is probably going to be fine, and that's one of the reasons that we're still vaccinating people in the UK with the JN1 vaccine. So I'm going to switch tack now and start talking about Ghana. And so at the beginning of the pandemic, I was introduced by Jean Langhorne, who helped with Gordon found the Crick Africa Network, and was introduced to Yao Bediaku. And Yao at that time was applying for some Gates money, a Calestus Dumas Fellowship, to do some COVID work as he just moved back to Ghana. And essentially what we were able to do uniquely is line up recruitment for our cohorts in parallel. So the heritage cohort that Yao would work as he just moved back to Ghana. And essentially what we were able to do line up recruitment in cohort that Yao identical schedule for the legacy cohort, particularly around the time of doses vaccine 1 and 2. So this provides a unique snapshot of what the humoral response and the cell-mediated response to SARS-CoV-2 looks like in Accra versus London. Similarly aged, different gender, different humoral response and the cell-mediated response to SARS-CoV-2 looks like in Accra versus London. Similarly aged, different gender, different occupations, but fascinating. And essentially this paper came out last year after a lot of work by together time course dose or infection outcompete legacy for every single variant that we tested, including ones that didn't exist yet. So the Ghanaian immune response was already primed at baseline and was consistently high. These people did not wane. Their antibody titers were high and stayed high throughout the duration of the study, despite only having had 2 doses of vaccine. So this led to an application to the Wellcome Trust for our WWW consortium, which brings in a further cohort in Jamaica, and this helps us ask this really fundamental question in biology: how much of what we see is due to environmental exposure and how much is due to underlying genetic variation? And particularly by being able to match up Ghana and Jamaica, we have 2 populations that are ancestrally very similar but geographically extremely different and have had different exposures. And it was shown very clearly that the mortality rate in the first year of the pandemic from COVID-19 in West Africa particularly was 10-fold lower than it was in Jamaica. So we've now, after a lot of work and a lot of logistics and a lot of organisation, been able to bring the 3 cohorts together and sync up simultaneous bleeds across 2024. Ghana saw lots of waves of infection, but then surveillance has fallen off, but we really haven't seen much. In Jamaica, they had slightly different but similar waves of infection, and then surveillance has fallen off. Ditto in the UK, these are all Omicron here, and surveillance has fallen off. Our vaccine history is different overall. Our legacy cohort are very highly vaccinated, mostly because they're scientists and healthcare workers and they actively seek out vaccines. Unlike Heritage and Windfall, where they were vaccinated at the beginning of the pandemic with 2 doses, but very little since. But we've been able to tease, unlike Heritage and Windfall, where they were vaccinated at the beginning of the pandemic with 2 doses, but very little since. But we've been able to here sync up all of our sampling, and we've essentially divided our sampling to try to control for wet versus dry, hot versus cold, well not hot in the UK, warm versus cold, because there's some seasonality that's inherent in vaccine virus transmission that we need to account for. And this is what we found. This is in preprint. This is not yet published under peer, so this is without peer review, so caveat. But essentially, what is really interesting is that our legacy cohort who've had more doses of vaccine containing ancestral Wuhan, unsurprisingly have more antibodies against ancestral virus. Not surprising, but when we get to the variants that circulated in 2024, the Ghanaians are neutralizing everything, along with the West Indians compared to the legacy cohort. So despite not having extra vaccines, they have significantly more abundant, these very high quality neutralizing antibodies against these variants that at the time of the bleed were just emerging. How does that match up across the 2 quarters? Well, what's interesting is that in this second half of 2024, lots of the legacy cohort had either an infection or a vaccine, and they catch up with the heritage and windfall cohorts. But they needed that significant boost to catch up. When we put that into antigenic space, we see that legacy here are 3 variants that we were interested in. They're quite separate, but when they get the vaccine, they cluster together. But look here, in windfall and heritage, they cluster already really closely together, and that doesn't change over time. This persistent, high-quality, durable humoral immune response, despite that very early vaccine exposure, is really challenging some of our notions of what's driving this immunity. How does it start? How do we drug it, essentially, for better vaccines? All right, so I'm nearly there. Oh. So this led to 2 obvious questions. What about other viruses? So first of all, we looked at seasonal coronaviruses, which we know circulate everywhere. And one of the questions in our WWW consortium is exposure in West Africa to other coronaviruses provide this cross-protective pre-existing immunity. Well, interestingly, we found again, the Ghanaians neutralize everything, more than the West Indians in case of OC43. And the legacy cohort do sort of catch up a bit, but there's still much higher antibody titers. Remember, this is a log 2 scale, so we're looking at really quite big changes on this figure. So, next questions that we're approaching now with WWW, and more generally, should we keep worrying about immune escape variants? We've been worrying about them for 5 years. Do we need to worry about them? Or actually, are we already pre-protected? Do we still need a correlate of protection? A correlate tells us in a point in time whether someone was protected against a point variant, and that's useful for some interesting biology, but do we need it to help us decide who's going to get another vaccine, who's going to get another drug, who's at risk? What is the underlying mechanism of this forward immunity? Have our germinal centres actually generated a whole load of redundant clones for B cells which are ready to pop out and produce antibodies at a moment's notice? Is this an evolutionary strategy that we've uncovered that actually has protected the human race against a lot of pathogens over a long time? And how does infection shape this durable immunity? Because you saw at the very beginning, and we see this consistently, that the biggest factor in terms of vaccine exposure that generates that an early boost, which then leads to that plateau, is an infection. And that obviously infection involves the mucosal space, it involves more T-cell immunity. There's lots of really interesting factors involved with infection in a human being versus a vaccine that we need to unpick some more. And that's something that Alicia and I are working on. And then finally, this fundamental question, what's the contribution of genetics or environmental exposure? One of the hypotheses we're working on in WWW is malaria exposure primes cross-reactive T cells that they can kill the virus stone dead. But it doesn't explain why everyone's got such high and persistent durable antibodies. So there's probably multiple mechanisms at play. So to wrap up, sequential exposures to SARS-CoV-2 enhances protective forward immune breadth. But predicting viral evolution is really complicated, more complicated than anyone ever thought. The role of mucosal immunity in shaping breadth is unclear. And the adults in Ghana, you can all pat yourself on the back, have preemptive neutralizing antibody breadth against all emerging variants despite limited vaccination. Isn't that cool? But why? We don't know, but watch this space. Mechanistic understanding of how infection shapes this forward immunity is crucial. So I'm going to wrap up with lots of people to thank. I'm not going to name everybody or list everyone because it's impossible, but I have a huge thanks to our partners here in Ghana, particularly at WACCBIP Yamachi and the University of Ghana, which has been a really productive and fruitful collaboration. I'm going to thank our funders, and I need to thank lots of people at the CRIC, but particularly leadership of the Legacy Study, Charlie Swanton, Sonia Gandhi, Steve Gamblin and Brian Williams, and I also really need to thank David Greenwood and Mary Wu, Ed Carr and Gift Azimbwe, who are my team, who we work very closely in partnership to generate all of these data and analyze them. And last slide. Oh my God, then. Thank you. Thank you for listening.
Thank you, Emma, for the fantastic keynote. I think we have time for a few questions. Sanka.
One thing that we have seen is in a group of cohort of patients who had infection during the Delta period, they got vaccinated after that. When we sampled them, the JN.1 was going on. And we tested their serum against the ancestral, the Delta and the JN.1. What we see is that they have truckloads of antibodies against the Delta, but very less against the ancestral, and to a greater extent against the JN.1. So it's like kind of the original antigenic sin kind of thing. Have you seen something similar?
No, we've seen the opposite. So we originally started with this hypothesis around original antigenic sin. And this is a concept that's arisen from the flu world, where people get specific flu exposures in childhood that then subsequently shape your B-cell repertoire and your antibody response when you see a vaccine. We're not seeing the same thing in our cohort, and we haven't seen it in Ghana, we haven't seen it in the West Indies, although the West Indies cohort was sampled later. But we've been following our cohort up, and with our even when we look at pre-vaccine and we look at those specific first infection exposures, we don't see that sort of really narrow. We expected to, and we didn't. And that's why people were going to CRIC leadership saying, I don't think Emma knows what she's doing because she keeps finding all these extra antibodies and they shouldn't exist. And so it's really, one of the ways we've been able to do this is because we've got such a high quality live virus platform, so we can really resolve down at really high level exactly the titers against different variants going forward. And we also genetically characterize the variants to make sure we haven't got any additional mutations involved in growing the virus in culture, which is a common problem. And we are starting to develop a pseudovirus platform, but we've found marked differences between pseudovirus neutralizing antibodies versus using live virus. So at the moment we continue to use live virus while we try and resolve those quality issues around the pseudovirus. And so it's something that needs more unpicking, and I think that the more people we can look at in different parts of the world who've had different exposures, I think we can start to answer these questions in a bit more detail.
Chinedu first, really?
His hand was up first.
Detail.
Chinedu first, really?
His hand was up first.
B cells, but it's hard choosing the right usual and unexpected, previously undescribed B cell population, and then part of that will involve sequencing the repertoires from those B cells. But it's hard choosing the right cells to sort that fallen out, stop neutralizing quite quickly, is sotrovimab. And sotrovimab was derived from a patient with SARS-CoV-1, not SARS-CoV-2. And so it's really broadly active and still continues to neutralize variants now, although at a much lower level, and they are starting to escape from that antibody. So that's a single antibody clone. So we know that there are broadly active reactive antibodies that react against everything, and subsetting is going to be really important.
Yes, following up from her question, you showed that the Ghanaian population reacted differently from the one you are studying. So if you did some BCR characterization, is there a way their repertoires were sort of having similar public clones that are not in your population?
Sorry, I don't get your question. You mean in the legacy or in the heritage cohort?
Yes, yes. So because the Ghanaian population, you say they continue to neutralize.
Yeah.
So how does that correlate?
Legacy or in the heritage cohort?
Yes, yes. So because the Ghanaian population, you say they continue to neutralize.
Yeah.
So how does that correlate with their repertoires? Are there like persistent clones? Okay.
That's something we're still trying to figure out how best to study, and part of that, and the most important bit is, as well as designing your experiment, is selecting exactly the right samples to go into that experiment. And one of my many problems, and the reason I'm so grey, is that we've been generating data so fast for legacy, for policymakers over time, that really now, when everything's calmed down, is the time we're really going back to some of these really fundamental questions and working out how do we address them. And it's one of the powers of WWW is that we've got these three cohorts where we can unpick those differences and really carefully select out the matched populations.
Excuse. Thank you very much for the presentation. I am Helen Nantambi from Uganda.
just an inquiry in the bid.
These three cohorts where we can unpick.
If it is to do with
antibody, it was a subset.
If it is to do with
antibody, it was a subset of neutralizing antibody in the fold, mean change titer, and convalescent against all of those original studies. It gets much harder to study now because everyone's antibody repertoires are so complex, and how do you pick out the binding and look at the FC effector function particularly, and how do you choose the right antibodies to do that? That's much more difficult. It is something we can go back to our early samples to start to unpick in a bit more detail, but we've been looking also at the functionality of the T cells, which are obviously really important. In vaccine, we know primes T cell repertoire. We also know that infection will prime the T cell repertoire, and how much of what we're seeing is actually specific antibody function versus actually highly effective T cells and B and T cells working together. And remember also that in the germinal center you need T follicular helper cells to make good antibodies, so you've got to have those T cells in there as well. So it's really complicated to try and unpick, and not something that's easy to unpick in a longitudinal cohort, and persuade a funder that actually it's worth going back to those original samples back from 2020 to try and answer these questions. But it is really important, I agree with you. You're going to wrap me up.
Oh, sorry. Absolute last question.
It's just a mundane question. How did you come up with these names, legacy, heritage, and windfall? And the windfall, is it out of Windrush?
No. So legacy is because it's a legacy of the testing pipeline, the research legacy of that testing endeavor that the CRIC ran. Heritage, Yao is here. He can comment in why he wanted to call that particular his cohort heritage.
Well, it was inspired to some extent by legacy, but we thought heritage, Yao heritage, it was our contribution to understanding COVID globally, but from that, and it was nice that legacy and heritage the words sort of fit together.
So, I need to say it again. I think everybody heard me, so yeah. Well, I'm giving a talk on Friday, so I can answer it on Friday. But no, I was just saying it was legacy. Heritage was an idea we had, or the project was an idea we developed sort of almost at the same time I met Emma. And in trying to name our study, we wanted a name that would somewhat match what legacy was doing. Heritage is something that we take very seriously in this country. And the goal of this study,
how Ghanaian science would also be globally relevant. And that would be a heritage that we use. So that's Windfall, I don't think
So Windfall was named by the Jamaicans, and it relates to the wind that comes through and hits the islands, those trade winds that came across and took all the boats across the Atlantic. It relates to the winds rather than the Windrush, which was the boat that took the first set of migrants from the Caribbean to England to help rebuild after World War II. Okay.
Thank you very much, Emma. Tough act to follow. If I try to summarize it, I think what Emma's talk shows us that as much as we know, the more you go into immunology, the more you find out, and the more you uncover, the more confused you become. So the next session, which will be the first of our plenary and then fellow sessions, will be chaired by the head of Department of Biochemistry, Cell and Molecular Biology, who was one of WACCBIP original staff, faculty, Professor Teresa Manfujira, and another OG, Professor Niels Ben Kwashi, and they'll take us through till the lunch break. So I'd like to invite them to come up to the table. All right.
Hello? Okay, it's working.
So I'm very privileged to be co-chairing with the head of the department. Her name has already been mentioned to you. So I will start off with the plenary section by introducing the speaker, and then she will take over after that. I looked at the topic for the section, and I seem to be